Welcome to MOOC NPTEL course on bioengineering and interface with biology and medicine. In today's class, we will study Mendelian genetics in more detail. We will discuss the chromosomal basis of inheritance and will find out what are the heritable factors involved as defined by the Mendel. I must say that Mendel made huge contributions for identifying that there are certain factors, heritable factors, which transmit from one to next generation. But quite long time people did not know that what those factors are. And then also people did not have too much information about whether those factors in which manner they are inherited. So few scientists started making more investigation, and one scientist who had made huge contribution in the area is Morgan, and I'll talk about some of his experiments. But before kind of what we are going to talk is, are the genes for the P properties which we are talking, are they present on the chromosomes, and do they segregate as a part of the divisions, meiotic divisions, the way we have been studying the meiosis process, right? So in this case here we have yellow and round, and we have green and wrinkled seeds. As a part of meiosis, these gametes are separated, and if these gametes contain the genes, those genes which are giving the properties like yellow and round seed or green or wrinkled seed, so are they also getting segregated as a part of these divisions which are happening? So genes are located on the chromosomes, and what is the behavior of these chromosomes during the meiosis process, and whether Mendel's law, which we have talked about the segregation and independent assortment, is also valid as a part of these chromosomal basis of separation. Little complex slide, but one could start thinking and refreshing yourself about meiotic cell division. So as a part of law of segregation, the two alleles for each of these genes will separate during the gamete formation, which you can follow the same what we have done in the meiotic process. And the law of independent assortment, it means alleles of genes on the non-homologous chromosomes would assort independently during the gamete formation. So it's very well color coded here in the red and blue, showing that your capital R and small r alleles for the same gene properties, how in the meiotic process they are getting segregated. So these are the kind of assumptions made, thinking about whether what Mendel has discussed, whether that has any basis at the chromosomal level. So far what we talked is essentially that Mendel's dihybrid crosses one could correlate also at looking at the behavior of the chromosomes during meiosis. During meiosis, these homologous chromosomes, they get segregated, and therefore alleles also get segregated. The behavior of chromosomes in meiosis process of the F1 generation, as well as after the fertilization in the F2 generation, could be observed by what Mendel has already shown in his crosses. So while these are some assumptions, but there is no experimental evidence that how one could prove this. And this is an area where I must say that another scientist who made huge contribution is Dr. Morgan, who studied the Mendelian inheritance and what are its physical basis at the chromosomal level. So this is a scientist for whom we are talking, Thomas Hunt Morgan, Columbia University, and he selected a model system which is Drosophila, or the fruit fly. So far we have been talking mainly about pea plants, and but at some point I had mentioned to you that many people like to work on Drosophila because again it comes in many properties. You can grow them very fast. I'm sure you would have seen Drosophila in some time on the banana peel. You will see those small flies with red eyes. And people make these cultures of these particular Drosophila stalks they make. This is showing you here lot of labs which works in the genetics and developmental biology area. They have the stalks of these Drosophila strains, and these Drosophila you can make mutations in different genes, and you can keep those and keep growing those, which can be very handy for doing lot of experiments and testing your hypothesis. So what is shown here, you have small tubes, and in those tubes you are having banana peels and some nutrients on which these flies can grow, and people store them and mate them and make sure that they are maintained well for doing a lot of genetic experiments. So this is where we are right now, but at that point in 19th century, Morgan started working on Drosophila and thought about looking at Mendelian genetics at the fly level. So let's see what kind of experiment he did. Finally, he provided evidences that chromosomes are indeed the location of Mendel's heritable factors. So I have, there is no suspense on that. I already made the conclusion. But now let's see the experimental system and what kind of things he did. So now nomenclature of the things, the way we have been discussing from the pea plant will slightly change. Here we are talking about phenotypes of Drosophila. Some characteristics, if these are commonly found, for example, red eyes, that is wild type character. And if a deviation of that is found, that is known as mutant type behavior. So if this particular eye is white eye, which is not commonly found in Drosophila, that is a mutant type behavior, whereas the red eye is commonly found normal phenotype, that is wild type. So I'm sure now you are familiar with the wild type and the mutant terminologies. Let's look at some of the how you can denote them. So far we are talking about in the pea plant context, only simple nomenclatures. Now let's add a little bit complexity here. When we say wild type, you are denoting with a plus sign, and when you have a mutant, then there is a no plus sign. We are also showing here male and female chromosomes. So this is a male Drosophila, so XY chromosomes, and X is having W suffix on that or superscript on that. So that is a mutant behavior. Now this particular thing is XX, a female. Both contain W+, that is a wild type, which will give the red eye pattern. Keep this in mind. I think there are some crosses which we have to do, which now we have to use these kind of nomenclature. So if in a given cross, some point, we'll talk to you about a given property, its wild type and the mutant behavior, you can denote those with plus or without plus. All right, so now let's see what kind of cross Morgan did. So he took these wild type female fruit fly as well as mutant white-eyed male fruit fly, and then observed their progenies in the F1 and F2 generation. So in this case, the female is red-eyed here and male is white-eyed in the parent generation. In the F1 generation, all the offsprings, they showed red eye. Just imagine what Mendel's experiment were, right? So purple was the dominant, and now that was shown in all the F1 generation. Something similar he also seen. Now he wanted to follow this particular cross in the F2 generation, and I would ask all of you now to start thinking and start making this kind of cross. So let's start doing that. All right, so we have the, so you have to now start making X and Y chromosomes. And then also you have to denote what is a wild type and what is mutant, right? Then only you can say that offsprings, how many are male and female and what are their characteristics. So let's try out simple, the same way of Punnett square. So let's draw only for F2 generation. So this is Y chromosome, and we have this X chromosome, which is having W+. All of you are doing that, right? All right, I'll come and see your results, all right? I'm sure right now it's not very difficult for you to do the crosses, but you have to assume that you have to know the terminologies, you have to know how genetic crosses can be done in different type of properties, which we will describe. All right, so I'm sure all of you have done your cross by now. So what is the phenotypic ratio? We are looking at one property, which is red eye versus white eye. White eye is the mutant property and red eye is the wild type property. And these signs are for male and female, which also is important to draw those. So it is not so difficult, right? Because you have done already these kind of crosses for Mendelian genetics in the P, and that time 3:1 were very simply defined. Yes. All right, so a good observation that whether we have no gene on the Y chromosome, and that is, I think, a right observation, that what Morgan also proved here, that certain genes, they are only carried on a specific type of either X chromosome or Y chromosome. So some properties are linked to specific sex chromosomes. They are not present in both the chromosomes. And therefore, and this observation did not come beforehand, it came only after this cross. So now if you realize, we have only one white-eyed male, right, which is the mutant type. So because he also observed 3:1 ratio, 3 other flies were red-eyed, 2 of them female and 1 male. But one of the white-eyed male, he was observing this only male. So based on that, when he made these crosses, he realized that the segregation is happening, but there is some sort of link to the chromosomal basis of that. Probably these genes are located on some specific chromosomes, X chromosome or Y chromosome, and that's why they are seeing this mutant behavior is coming only in the male type here. So I think it's important in genetics that you should make those observations for a given cross and then use all the properties, label properly, and then probably you cannot make any mistake. So this is kind of cross which we just talked. We have these gametes from the sperm, W plus and the Y chromosome here. From the X we have W plus and W. And now if you are making these crosses, you have only one white-eyed male. You have remaining 3 flies which are all red-eyed. Yes. So phenotype-wise, there 3 are red-eyed. Phenotype, if you look at of the 4, it's 3:1. All right. So what we just discussed, the cross. The main points of this cross are that the mutant or the white-eye trait, that is recessive as compared to the wild type of the red eye trait, which is W plus. In the recessive trait, white eye was expressed only in the male in the F2 generation. I think you had rightly observed this, and that led Morgan to conclude that eye color gene is located on the X chromosome and not on the Y chromosome. So while that's segregating, it is some specificity that where these genes are located. So conclusion from the experiment is, we saw the evidence that chromosomes are indeed the locations of what Mendel has defined, those heritable factors. So Mendel was only able to say that there are some factors which are transmitting from one to the next generation. Morgan was also able to show that there are some chromosomal basis to them. All right, so genotypic ratio, you can say in the female what is the genotypic ratio, in the male what is genotypic ratio, and overall genotypic ratio of all the offsprings. So in that way, yeah, it can be, you can say that among the female this is the genotypic ratio, among the male this is genotypic ratio, among all of the 4 what is the phenotypic ratio, or even you can split into male and female and you can put in the ratio form. So if you think about human and the chromosomal basis of sex in human, we have 44 pair of the 44 autosomes, and XY chromosomes are there. So these 22 plus X or 22 plus Y part of the sperm and 22 plus X part of the egg. This is known as the XY system, where X and Y chromosomes and the various genes which are linked to them are getting segregated. So, sex of a child actually depends on whether the sperm contains X or the Y chromosome, because egg will always carry X chromosome only. So, the sex of child will be derived from the sperm, whether it's coming from the X part or the Y. So, there are many sex linked genes which are only found on the X chromosome or the Y chromosomes, and the Y linked genes are actually much smaller in number. Only around 78 genes have been found on the Y chromosomes, whereas on the X chromosome almost 1100 genes are there. So, a lot of properties are only uniquely present on the X chromosome or on the Y chromosome, and they will follow that pattern coming from these chromosomes. If you look at this circle, can you read something in that? How many people cannot read? Cannot read? How many people cannot read? All right. So, well, if in case you are not able to read, I think you should pay some close attention to this, and you should go for some test. You might be not able to distinguish the colors so easily, and this is reality, right? Because for many of the exams, especially for the railways, military, etc., they also test for your color blindness. Can you distinguish the colors clearly? So many of the disorders which are sex linked includes red-green color blindness, muscle dystrophy, you have night blindness and hemophilia. So many of these diseases are actually derived based on the transmission of X-linked recessive traits, especially for the red-green color blindness. So let's think about the red-green color blindness, which is much more apparent case. Let's think about again some of the ways to denote the conditions. Please pay attention to this slide. A lot of questions are going to be based on this. So for a color blindness to happen, this gene is going to be on the recessive allele, which we are denoting from the small n. If an individual is unaffected or normal eye, normal vision, then we are showing from the this white square. If it is carrier, we are showing from the light shade. If color blind, from the dark shade. So this case, let's think about female and male. A cross is happening. In this case, it is normal. When we have normal phenotype, it means capital N and capital N, right, on both the chromosomes. And if it is carrying the recessive allele, which is small n, then only you have the disorder. So if I am just showing you this, what is this cross is between what type of male and female? This male is
colorblind, yeah.
And female?
Female is unaffected.
All right. So female is unaffected and male is colorblind. All right. Now let's take second situation. In this case, what will happen? What female is?
Carrier.
Yes. And male?
Unaffected.
Male is unaffected and female is carrier. Let's take the third situation.
Female is carrier and male is colorblind.
All right. So I'm hoping that in addition to looking at just the colors, you're also paying attention here that whenever there is a small n, which is recessive allele is there, with the male, it has to be only one on the X chromosome. So it's always getting color blind. Now in the female, even if it is present in one copy, then still it is only carrier, it is not showing the full effect, right? Now let's try to take these conditions and then do the crosses and see what will be the fate of these offsprings coming from these crosses. Please start doing that. So that's the first situation we had talked. Second one is that we have male which is normal and female is carrier. Third cross, we have male him colorblind and female is the carrier. All right, all of you are doing it, right? Also should write based on the X, 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 Y, which are male and female. All right, so I'm sure all of you have done the cross as well. So you have to now do these crosses and give me the answers for each of the cross. What will be the progenies look like? And when the questions will be asked, you will be actually given a situation when that, if the cross happens and the kind of child which are born, 50% of them are carrier and 50% of them are normal, what could be the genotype of the parents? So then you have to go backwards. So let's go one at a time. The first cross, is everybody trying now? Everybody is comfortable? How about this side? Okay, so let's think about different crosses which we have done. The first situation is when we have a color-blind male and we have the normal female. From this, the progeny which are born, of them 50% of them are carrier. Okay, you will not see apparent any change in the phenotype, but the 50% of the child are actually going to be carrier. So a color-blind father is going to transmit these alleles to their daughter. It's only transmitting, and it is not going to be apparent in the next generation. Second situation, the second cross, we have the female which is carrier, and now male is normal. In this case, out of the 4 progeny, there is a probability that one of the male child will be colorblind and one of the female will be the carrier. Now let's think about the third situation, when we have a colorblind father and mother is a carrier. In this situation, you have 50% child who are going to be colorblind and 25% of them will be carrier. So many times if a family has the history of carrying these kind of disorders, so it's a good idea for them to do genetic testing or get genome counseling before marriages, because you want to know that what could be the fate of those small genes and whether they will be apparent in the progeny. And therefore, many people make those kind of choices. In this case, if let's imagine here, in this case, even if the father is actually color blind, but as long as mother is totally normal and not carrier, then the progeny will not have any effect. But if this is the third situation is there, the father is color blind and they are not doing any genetic testing, they are marrying with anybody, and then that female is also carrier, then the possibility is that most of the child will be either color blind or they will be carrier. So these are reality, and many times when these genetic disorders happen, people do these genetic testing to ensure that they can avoid this situation. Okay, so these things can be just done in the same kind of mathematical probabilities the way you read many other mathematics questions. So conclusions from these crosses are that fathers pass X-linked alleles to all of their daughters, but none to their sons. And these child daughters, they are actually carriers. They are going to carry from that generation to the next generation. Mothers can pass the X-linked genes and alleles to both the son and daughters. Any male which receives the recessive allele from the mother will express the trait. Sometime I'll just give you this kind of statement, and I'll ask you to show this is true or false by doing a cross. The small cross which I showed you are very simple, but it becomes difficult when you have been just given a statement and you have to derive the crosses based on that. So I am hoping that from this exercise you are getting more comfortable now, and you should be very easily able to derive those crosses now. So far more males than females actually have X-linked recessive disorders. So absence of one or more of the proteins which is involved in doing the blood clotting is not found in this particular disease condition, and therefore there is excessive blood loss happens from the wound site. In fact, nowadays, because of all our advancement understanding of doing genetic engineering and protein research, it is now possible that we have those clotting factors which in the purified form present, which could be injected in the individuals, and one could try to overcome this deficiency. But of course, that time it was much more serious issue. And what was more interesting, that people started observing Queen Victoria's family, that many of their descendants were actually passing these traits. So many of the royal family children were carrying these alleles, and therefore some of them started showing these symptoms, and then it became more apparent that it's not only one child which is having problem. There are many from that family who are having these issues, because their intermarriages were from the very small group of individuals from Spain, Russia, etc. But this was definitely one of the very interesting fact. It was happening in royal family, so it became much more apparent, and everybody started noticing this problem. And that time some scientists published a study on hemophilia, and then they this thing became much more like a case study for all the scientific community to find out what is happening in the royal family and how the genes are getting inherited. Okay, so the conclusion is that you may now overcome some of these deficiencies because we have understanding of how to synthesize these proteins and the clotting factors, which could be injected in the individual, and that may prevent the blood loss. But nevertheless, understanding the genetic basis is very crucial, and these kind of genetic counseling per se can actually help us to avoid many of these kind of mishappenings. So by now you are familiar with the chromosomal basis of inheritance. You now know that Mendelian inheritance has its physical basis in behavior of chromosomes. It is the behavior of chromosomes during meiosis that accounts for Mendel's law of segregation and independent assortment. Morgan independently tested Mendel's experiment and provided evidence that chromosomes are indeed the location of Mendel's heritable factors. You also studied sex-linked inheritance and how Y-linked genes mainly help determine sex, while X-linked genes help in determining genes for many characteristics, but unrelated to the sex. In the next class, we will study genetic recombination and linkage, which is very important in determining the inheritance of characters. See you in the next class. Thank you.